Welcome to Amoeba Classes, my dear students. In this lecture, we will be studying about cell division and the structure of chromosome. This lecture is based on your class 10th ICSE board syllabus. This lecture that I am going to discuss will be in different parts, and this is the very first part of the lecture. According to your ICSE, the very first chapter is cell division and the structure of chromosomes. So we will be starting from the very first chapter step by step. So before starting, let's just look at the syllabus, what the ICSE class 10th 2021 syllabus is, and then we will be continuing. Talking about the syllabus, so here is the screenshot from your scope of syllabus, and if you want to take a look at your scope of syllabus, you can download it from the website given in the description below. Okay, so if we take a look, the very first chapter or the very first unit is basic biology, in which these two chapters, the very first chapters are these, that is cell cycle and cell division. The another one is the structure of chromosomes. Now, before starting cell cycle and cell division, we should be knowing that what is chromosome and how does the structure of chromosome looks like. In your class 9th, if we talk about your class 9th, in the very first chapter, you have already studied about the structure of nucleus and the chromosomes, the chromatin fibers. If you don't remember anything, you can move at the top right corner over here. You will be clicking the I button if you are viewing this video, watching this video on YouTube. Click on that video that is from class 9 chapter number 1, and you can skip to the timing 7 minutes 32 seconds. Okay, and from there the video will begin and you will be getting a little bit idea about the structure of chromosome that was in your class 9th. In your class 10th, we have to study about the basic structure of chromosome with elementary understanding of the terms, right, such as chromatin. We should be knowing, right, that what is chromatin basically, what is chromatid, the gene structure of DNA. That means we have to learn the structure of DNA, that what it is basically, and the part centromere. So 1, 2, 3 and 4. These 4 things we have to understand very properly, so that we can begin with the very main content of the chapter, that is cell division and the cell cycle. Okay, after completing this structure of chromosome, we will be starting with the cell cycle and cell division. So in this we will be studying about interphase, G1S, G2, mitotic phase, and the cell division, mitosis and meiosis. The stages are not required, and homologous chromosome. This homologous chromosome we will be discussing in this topic only in the same video. Crossing over, separate video it will be. Significance and the major difference between mitosis and meiosis. Okay, so this video will be in different parts, and in this very first part, in this same video, we will be discussing about this topic along with the homologous chromosomes. So let's begin with the structure of chromosomes. So first of all, we should be knowing that where are these chromosomes located. So as you remember that this is the structure of a cell, and inside the cell there is a nucleus, right? There is a nucleus, and inside the nucleus, if we talk about, there are chromosomes present. Now if we take a look at the structure of chromosome, this is how the chromosome looks like. And if you have seen the video of class 9 that I have recommended, now you will be understanding that what is basically a chromatin fiber. Chromatin fiber is basically the structure like this, the thread like structure, the network of the thread like structure like this, and when this chromatin fiber condense together, and after condensing it forms the structure like this, and this is basically what? Chromosome. That means chromatin fiber after condensation forms what? Chromosome, right? Now if we decondense, or if we uncoil this chromatin fiber, if we uncoil it, right, what we will be getting? We will be getting the strands like this. Okay, suppose this is a chromatin fiber, and if we zoom it out, we will be seeing some structures like this. And what it is, this is what? DNA. You know the full form, right? If you know the full form, comment down below and I'll be checking out. Now if you take a close look over here, this structure, if you take a proper look, this DNA that I have made over here, this part, this DNA, it is coiled around a structure that is these proteins, and these proteins are called as histones. Okay, what are these proteins called? These are called as histones. So there are total 8 histones. Okay, there are total 8 histones protein. This histones protein we will be learning at the very end of this video, but right now just understand that this is a DNA, and this DNA is coiled around the histones protein. There are total 8 histones protein, and these histone proteins helps in the packaging of the DNA, and together along with the DNA and the histone proteins we call it as nucleosome. This we will be studying at the very end, the last topic in the same video, so don't worry about it. Okay. Now if we take a look at the structure of chromosome from another perspective, so over here you can see this is the structure of chromosome. Now let's understand some basic things. This is a chromosome, right? If I make a proper diagram over here, let me draw the diagram. Okay, so this is about similar. Now the very center part that you see, this is called as centromere. That means this part, this circular part or spherical part, this is the centromere. At the top you can see 2 arms over here. Okay, each single arm is called as chromatid. What it is called? Chromatid, right? So this is one chromatid and this is the second chromatid. Okay, don't get confused that it is 1, 2 and this is 3 and this is 4. No, this is totally wrong. This is not third and the fourth, so I'll be removing this 3 and 4. So this whole thing like this is the one chromatid, and this whole another one is the another chromatid. That means how many chromatids in this chromosome? 2 chromatids are there. Now in the centromere there is some structure like this originated from the centromere, and this is called as kinetochore. What it is called? Kinetochore. This is called as kinetochore, and this kinetochore is a place from where the spindle fibers joins during the cell division. We will be discussing in the next video. Okay. So over here you can see the centromere is written over here, the spindle fibers during the cell division, right? It attaches to the centromere. That is basically what? Kinetochore. Okay, so I hope you understood the structure of the chromosome. Talking about the chromosome, we know that each cell contains how many chromosomes? 46 chromosomes. How many chromosomes it contains? 46 chromosome, and in pairs how many it will be? 23 pairs, right? Now what is basically this 23 pairs? There is one term that is called as diploid. What it is called? Diploid. Diploid means 2n, and what does 2n stands for? It means 46. That means whatever the total number of chromosome in any organism, that is a diploid value. Suppose if I tell about any organism, suppose there is an organism called as X, and if I say the X contains total 1000 chromosomes. How many? 1000 chromosomes. That means the diploid value of this organism X will be 2n is equals to 1000. Understood? So this is what basically the diploid value. Now if we half it, right, if we divide it by 2, we get another value that is called as haploid. And according to the basic mathematics, if we want to get the value of n, so what we'll be doing, we will be dividing this 46 by 2. So n is equal to 23. That means whatever the half number of chromosome value, it is the haploid value. If we talk about this organism X, what is the haploid value? 500. Understood? That means we have 46 chromosome. Our diploid value, that is our 2n value, is 46. Our haploid value is 23. Suppose if the question comes, what is the triploid value according to this number of chromosomes? So triploid is basically 3n. That means what you will be doing, what is the value of n? 23. So 23 multiplied by 3 will be your answer. Understood? So this is how it goes: haploid, then diploid, then triploid, then tetraploid, and so on. So right now you don't have to worry about anything. Just make sure that you remember these two things: haploid and diploid. Our haploid value is 23 and our diploid value is 46. This haploid and diploid terms, these are very, very important in the future discussion when we will be discussing about the cell division. Now this topic, the types of chromosome or the features of different types of chromosome, this is not actually in your scope of syllabus because it's not written, but according to me, I think this is a very important topic that you should be knowing. So we will be discussing it with a little fast speed, so make sure that you pay proper attention. Now as you remember, right now we have discussed that what is centromere, right? The center part of the chromosome. This is the centromere, right? Now let's understand this. This is a centromere and this is the chromosome, right? Now this is called as the P arm and this is called as the Q arm. Okay, got it? And this is the centromere. Now we'll be starting from the very fourth one. The fourth one is metacentric. What does it mean? That the centromere is present right in the center. So both the arms are equal. That means in metacentric what happens? P arm is equal to Q arm. That means this centromere lies in the very, very center and both these arms are equal in length. The next one is submetacentric. What happens in submetacentric? Centromere is slightly displaced from the center. Okay, these are the keywords. Centromere is slightly displaced from the center. That means one arm will be bigger. So suppose this is the centromere and that means one arm will be bigger. So this one arm is bigger and this one arm is a little bit smaller. If we compare it from this one, this Q arm is bigger, this P arm is a little bit smaller. That means slightly displaced from the center. Who is displaced? Centromere. So we get two unequal arms. The third one is acrocentric. Centromere is present near the extreme end. Okay, near the extreme end, not at the extreme end. It is present near the extreme end, and because of which the P arm is very, very small and the Q arm is very, very long. So make sure to remember this. And the last one is telocentric, and the centromere is present at the proximal end, and if the centromere is present at the proximal end, the P arm will be absent. That means only Q arm is present. And if we talk about this telocentric type of chromosome is absent in humans. That means out of those 46 chromosomes that we have, none of them is telocentric. Make sure to remember all these four types of chromosomes, and these are classified on the position or on the basis of the position of centromere. So make sure that you don't get confused. Okay, now on the screen that you are seeing right now, there are two images basically. One is this one and the another one is this one. Okay, these are the chromosomes. Okay, these are seen under the very, very special microscope. Okay, and this image I have downloaded from the internet. This photo that you are seeing right now, in my college days I used to get these type of photos where we have to identify all the 46 chromosomes and their type. Okay, so if I zoom out in this photo, let's try to identify. Okay, these are some extra knowledge that I am giving right now. If we see clearly in this chromosome, the centromere is the present and these two arms are equal. That means it is what? A metacentric chromosome. Now as we know that there are 23 pairs, that means each pair is similar, right? It is homologous. This homologous word we will be discussing afterwards. That means the same metacentric with the same size, it should be somewhere else also. So if we look over here, right over here, we found it is metacentric. Okay, I have already numbered it before during my college days, so it will be very easy for us to discuss. We will not be discussing all the 23, so don't worry, just for the reference. So this is what the metacentric. Now if we want to search the second number, right? So this is the second over here. Again this is meta, but the size is a little bit small. Both the arms are equal, centromere is in the center. And if we see the another number 2, yes, we see over here the number 2. Again this is what? Metacentric. So like this we have, if we talk about, this is acrocentric, this is submetacentric, right? And we have seen metacentric also, but nowhere over here in this image you will find telocentric. Why? Because telocentric is absent. Now if we want to take a look that where are the X and the Y chromosomes, so this is the Y chromosome. Y chromosome is basically acrocentric. Okay, so this is the acrocentric chromosome over here, and if Y is there somewhere, X will be also there. Let's try to take a look, I guess over here, right? So this is submetacentric X chromosome. Okay, it is bigger in size as comparatively to Y. Y is the smallest chromosomes. Okay, now what we have to do in this, actually in my college days, we have to cut these chromosomes with a pair of scissors and we have to stick on the separate page. Okay, so after cutting it out and sticking to the separate page, the image we get is like this. So this is the first pair of chromosomes, second pair, third pair, fourth, fifth, 6, 7, 8, 9, 10, 11, 12, 15, 16, 17, 18, 19, 20, 21, 22. And the last one is the X and Y. So like this we have to arrange these chromosomes in the proper order, and this arrangement of the chromosome in the decreasing order of the shape and the size, this property is called as karyotype. What it is called? Karyotype. So the image that you are seeing right now, it is basically what? Karyotype. So make sure to remember this simple stuff, and I hope you will not forget. And if you're planning to join or taking biology in class 11th and 12th and you are going to the college, I hope you will be learning about these things in the future. Let's talk about the structure of the DNA. As we have discussed in a rough manner that the DNA is somewhat like this. So this structure is basically the double helix structure of DNA. Okay, so it is what? A double helical or double helix structure. Right now, what is this DNA basically made up of? Okay, let me do a quick revision. This is your what? Chromosome. When we decondense it, we get what? Chromatin fiber. And if we zoom this chromatin fiber, we get what? DNA, right? If you remember, we have discussed in the very beginning of the chapter. And in this DNA there are some histones proteins that are coiled around. How many histones are there? Total 8 histones protein, right? Octamer, that means 8. Total 8 histones protein are there and they coil around it, and that means histones are the proteins that help in the coiling and packaging of DNA into structural units, and these structural units are called what? Nucleosomes. Very important, this nucleosome, make sure to remember. Okay, so this was the part about nucleosome. Okay, so if you want to read it out what is given over here, you can always pause the video and read it, take your own time, or you can take a screenshot also and you can read it later. So in this structure of the DNA, you can see over here there are 2 strands, right? We are talking about it as a double helix. So why double? Because 2 strands are there. First strand is this, second is this. So this is going like this in a helical form, and this is also in a helical form. If we unwind it properly, so it will be the straight lines, but these straight lines are anti-parallel. That means one strand, if it is going in the upward direction, another one is going in the downward direction. Now these two strands are connected by some bonds, and these bonds we will be discussing it right now. These bonds help in the combining of these two strands, so the stability of the DNA is maintained. Now to understand the exact structure of DNA, we have to take a look at this diagram. Now see, this is one strand, this is another strand. Now each strand is made up of some chemicals, right? And now what are those? The strand A, it is made up of sugar, and this is pentose sugar. That means it is made up of 5 carbon. Okay, 1 2 3 4 and 5. This carbon number and the structure you will be discussing or learning in class 10th chapter organic chemistry, that is in chemistry subject. Okay, so don't worry right now. So this is basically sugar. The sugar is attached by the phosphate group, so I'm representing it by P, and then we have the base, and these are basically nitrogenous bases. Okay, now first of all we have to understand what this nitrogenous base is. So before beginning we should learn some new things. Now there is a term called as nucleotide. Now what is nucleotide? Nucleotide generally contains phosphate group, it contains sugar, and it contains nitrogenous bases. Okay, now what is nitrogenous base? Nitrogenous base are some chemicals, so we will be discussing their structural form, right? So nitrogenous bases are basically of two types. One is called as purines and another one is called as pyrimidines. Okay, purines and pyrimidines. In purines there are two nitrogenous base, one is A and another one is G. In pyrimidines there are again two nitrogenous base, one is C and one is T. Okay, now if you know a little bit more about this pyrimidines, just for the extra knowledge, there is one more pyrimidine, and that is that pyrimidine is U. Okay, and this U is basically found in RNA, not in DNA. And right now we are discussing about the DNA, so we have only 4: A, G, C and T. A is adenine. Okay, adenine, guanine, cytosine and thymine. Okay, so don't worry about the spellings. So here it is: adenine, guanine, cytosine and thymine. Okay, so these are the nitrogenous bases. Now this is one strand, this is another strand. Over here, here is the nitrogenous base and here is the nitrogenous base. Now these two nitrogenous base, these are connected by some bonds, and these bonds are hydrogen bonds. What are they? These are the hydrogen bonds. Okay, not James Bond, not any other bond, not Fevi bond. Make sure to remember that these are hydrogen bonds. Okay, jokes apart, make sure to remember hydrogen bonds, very very important. Now these nitrogenous bases pair with each other. This adenine, it always pair with thymine. The guanine always pairs with cytosine. The adenine always pairs with thymine by double hydrogen bond. Okay, and the guanine pairs with cytosine by the triple hydrogen bonds. Okay, so this is the double hydrogen bond, this is the triple hydrogen bond. Make sure to remember adenine double bond T, right? G double bond C, and these bonds are hydrogen bonds. This can be vice versa also. Thymine can be paired with the adenine by the double hydrogen bond. Okay, by the double hydrogen bond, and in the same way cytosine can be paired with guanine by the triple hydrogen bonds. Okay, so make sure that you don't get confused in these terms: adenine, guanine, cytosine and thymine. Okay, adenine by thymine, by double hydrogen bonds. These are the hydrogen bonds, and guanine with cytosine by 3 hydrogen bonds. Okay, so the structure now see over here. Now you will be understanding it very properly. This is what phosphate, this is what sugar. Now after comes the nitrogenous base, and over here the nitrogenous base is T, thymine, right? The nitrogenous base is T, thymine, and thymine is bonded by double hydrogen bond with the adenine, and again adenine is bonded by the sugar, and the sugar is bonded by the phosphate. Now why this is going down and this is going up? Because we have discussed these two strands of DNA are anti-parallel. That means one strand is going down, another will be going up. Okay, this is going up and this is going down. That means they are anti-parallel. So over here the phosphate is at the top, and over here the phosphate is at the very bottom. Now this together, this nitrogenous base, sugar and phosphate, what it is called? Nucleotide. Now if you will look at this diagram, this is what? This is a single nucleotide, the basic structure of a nucleotide. So this is a nucleotide and this is a nucleotide. That means the DNA consists the two chains of nucleotide, one going upward, one going downward. And because this is the first nucleotide, this is the second nucleotide, this is the third nucleotide, and continuously the nucleotides are arranged just below each other, so this long chain of DNA, it is termed as polynucleotide. What it is called? Polynucleotide. Got it? So in the same way, the cytosine is bonded by guanine by triple hydrogen bond, and again sugar and phosphate. Now if we look properly, phosphate, sugar, thymine, adenine, sugar, phosphate, okay, double bond. Now this is one pair of nucleotide, right? This is another one. Now this nucleotide, this whole one set of nucleotide and another set of nucleotide, these again are bonded by a special type of bond that are found over here. That means it is joining what? It is joining phosphate, and this is the sugar and this is the sugar. Okay, so sugar, phosphate, and this sugar is of the another nucleotide. So over here there is a special bond, and this special bond is called as, this special bond is called what? It is called as phosphodiester bond. Okay, phosphodiester bond. That means the two chains of nucleotide are bonded by hydrogen bond, and the polynucleotide chain is bonded one below the other by the bond that is the phosphodiester bond. So make sure that you remember these things. Okay, so everything is written over here. You can pause the video and read it. Histone proteins we have already discussed. Okay, some points you can read it by your own, and I hope I have discussed everything. Some basic things are there, right? If we talk about the chromatin, it contains DNA and histones. DNA is deoxyribonucleic acid, 40% it is, and histones are 60%. And make sure to remember the function of histone proteins. They help in the coiling and packaging of DNA into the structural units. And what are that structural unit called? That is called as nucleosome. So I hope you understood till here. Okay, the chapter, this video is still not finished. Another topic is left, so make sure that you hold it. Before understanding the actual cell division, you should be knowing some terms or the definitions like diploid, haploid. We have already discussed this diploid and haploid, right? But we will be discussing it more. Then homologous chromosomes, centromere we have discussed, so we will not be discussing. Chromatid also we have discussed, so we will not be discussing. So let's take a look at this diagram and see what they have given. Okay, now in this cell you can see how many chromosomes are there. 1, 2, 3 and 4. That means total there are 4 chromosomes. So the total number is 4. That means it is a diploid value. Understood? So 2n is equals to over here is 4. As I've given the example of X, that the total number of chromosomes in the X is 1000, so it was its diploid value, and the haploid value was 500. In the same way, the total number of chromosomes are 4. Right, they have given 2 pairs or 2 sets of chromosome, but the total number of chromosome is 4. That means the haploid value will be 2, right? So if you will see in this diagram, how many chromosomes are there? 2. That means this is the haploid number. Okay, we will always be comparing. Make sure that you compare it properly. We are comparing with this diagram. Okay, over here 4 chromosomes and here 2 chromosomes. I don't know why this pen is disturbing me a lot. Okay, now to understand the homologous pair of chromosome, let's read out what's written over here and then we will be discussing. Homologous chromosome, it refers to the pair. Make sure first of all the very important word, pair of chromosomes, right? In a diploid cell, pair, you know, diploid cell, you know, that are exactly similar. Okay, they are saying similar, it is not written identical. There is a difference between similarity and identical. Make sure to understand this. Similar is something different, identical is something different. Okay, so it is similar in shape, size, and have the centromere at the same location. Talking about the location, the term this homo means same, logous means loci, and loci means location. So homologous chromosomes are those chromosomes which are similar in shape and size, and the centromere is located at the same loci, that means same position. They have genes for the same trait. Trait generally character, I'll be telling you within 1 minute. Trait is generally the character. In each pair, one of the chromosome is inherited from mother. The chromosome of the mother that we have inherited, that is called as maternal, okay, by the egg cell, and one from the father by the sperm cell. Okay, so talking about this, each pair, one of the chromosome is inherited from the mother, from the egg, and from the father by the sperm cell. A human cell consists of 23 pairs of homologous chromosomes. Okay, so if you haven't understood, don't worry, let's begin the understanding very properly. Now see, we know that we have how many chromosomes? 23 pairs of chromosomes. As the book is saying that all these 23 pairs are homologous chromosomes, right? Homologous chromosomes are those set of chromosomes which are having the similar structure and size, similar shape and size, and they lie on the same loci, that is the position. Now understand, 23 pairs, if we separate these 23 pairs, we get 22 pairs and we get 1 pair. This 22 pairs of chromosome, these are called autosomes. What are they called? Autosomes. And this 1 pair of chromosome, these are called sex chromosomes. Now in the sex chromosomes, again this pen is disturbing. Okay, so in the sex chromosome, if we talk about, we have 2 gender, right? One is the male, another one is the female. So if you don't know the sign convention for the male and the female, this stands for male. How you have to draw a circle and the arrow like this. For the female, you have to draw the circle and plus sign like this. Okay, so this is the male and another one is female. If we talk about in the male, we have a germinal cell, that is the sperm. What it is called? Sperm. And in the female, the germinal cell is called as ova. In class 8th reproductive system, I hope you remember there, the animals, the human beings, the living organisms, if it is male and female, the male will be having the male gamete, and that is the sperm, and female will be having the female gamete, that is the ova. Okay, so this sperm is haploid. Sperm is what? Haploid. And ova is also what? Haploid. That means sperm and ova both are haploid in nature. And if we talk about this autosomes, these autosomes, they are diploid in nature. Okay, now how these chromosomes are arranged. See, in the karyotype we have studied, let me draw first. This is chromosome and this is chromosome. Okay, so for example, this is the first pair of chromosome that we have seen in the karyotype, right? Metacentric, equal arms it is having. Now, as the book is saying, one chromosome is coming from the mother and one is coming from the father. That means in us we are having, this is one pair, so we will be just representing this. Suppose this is paternal and this is maternal, from the father and from the mother. Now, what actually is homologous chromosome? First of all, they are the pair of chromosome in which the centromere lies at the exact position. That means the centromere is at the very center. So okay, now the chromosome contains genes, right? Genes are the short segment of DNA. DNA we have already discussed. Suppose I am making some genes 1, 2, 3, 4, 5. So at the exact same position there will be 1, 2, 3, 4, 5. Got it? Now if we talk about the father, suppose there is one character. If we take one character and if I take character eye color, okay, now there are many different types of eye colors, like the very basic one is the brown and the black. Okay, so we will take example of the brown eyes, and instead of black we will be taking blue color, right? If you remember some celebrities, Zac Efron, Aishwarya Rai, they have the blue eye color. In eye chapter only you will be learning why there are different color of the iris or the eye. Okay, so brown and the blue. Suppose this is the character, okay, and this character is representing what? Eye color. So at the same loci, same position, both will be at the same position, and these both the genes will be representing the same character, that is the eye color. But what is the catch over here? Father, if it is having the eye color, this gene will be carrying which eye color? Suppose we are taking brown, and the mother is having which eye color? The blue color. That means these chromosomes are similar. Okay, these are similar, but they are not identical, because if they were identical, if brown is over here, in the same way brown should be over here also, but what it is? Blue. That means these are the pair of homologous chromosome which are of similar shape and size, and in which the centromere lies at the exact same position, and the genes are also present at the same location, and these pair of chromosome, they are similar but not identical, and these chromosomes carry character in the form of the gene which shows different characters and expression. Okay, it can be the height, it can be the eye color, it can be the hair color, it can be the shape of the nose, the shape of the ear. Okay, all the character that is present in the human body, everything who controls the genes. Okay, so this was the study about the homologous chromosome. So over here if you see homologous pairs in the diploid cell, so this is one pair, this is the another pair. So these are similar in shape and size, but they are non-identical. They are similar but non-identical. Okay, and over here centromere, this is the centromere, right, and both are chromosomes actually. Okay, in the next lecture during the cell division I will be making you understand that why this is also called as chromosome and why this is also called as chromosome. Okay, why in the other lecture, let's discuss in the same lecture only. See, this is also a chromosome and this is also a chromosome. This chromosome is having one chromatid. How many chromatid? One chromatid. And this chromosome is having how many chromatid? Two chromatids. Okay, we can count the number of chromosome on the basis of the centromere. Both the chromosomes are having only one centromere, that is, this is also a chromosome, this is also chromosome. But what is the difference? This chromosome is having only one chromatid, and if we talk about this one, this chromosome is having two chromatids. Okay, uh, over here it is the diagram over here. These are the chromatids. How many chromatids? Two chromatid. One chromatid this, another chromatid is this, and this is a single chromosome, right? And this is also a single chromosome, but it is having one chromatid. Okay, so make sure that you don't get confused. So I hope you understand all these type of things, and you can always pause the video to take a screenshot and you can read it later. So this was the very first video. In the next video I will be directly starting with the cell division. Cell division, very important topic as well as the chapter, and many questions in the board exam come from the cell division chapter. So I hope you all understood very properly and clearly. I've tried to explain each and everything, and if anything is skipped or if you have any doubt, you can always comment and I will be trying my best to answer you over there. Okay, so make sure to subscribe this channel if you have not yet subscribed. Share with your friends, your family members, your relatives, your brother and sister, those who are in class 9th, 10th, 11th and 12th, and make sure to recommend them this channel, the Amoeba Classes. Okay, so the next video will be uploading soon, maybe tomorrow or the after tomorrow. So I hope you will watch this whole video. Make sure that you watch whole video. If you will forward it, so you will not be able to understand it properly. So I guess it's time to take a goodbye. So let me write it down there. Good and bye. So goodbye, wish you all the very best. Make sure to learn it properly, and I hope you all are enjoying in this lockdown period. So play games, have fun in this lockdown, but make sure that you don't forget to study. Okay, so bye bye.